Welcome to today's deep dive. We're really thrilled you're here with us. Today, we are taking you to the absolute cutting edge of virology.
Yeah, we're getting into some truly groundbreaking territory today.
Well, we really are. We're unpacking a highly dense, totally fascinating 2026 paper from the journal Cell. And the subject is the Andes virus. Now, if you haven't heard of it, this isn't your run-of-the-mill seasonal bug.
Not at all.
Andes is a New World hantavirus found primarily in South America, and frankly, it is terrifying. It typically spreads through the air from rodent droppings, but uniquely for hantaviruses, it can actually spread from person to person.
Which is exactly what makes it such a massive public health puzzle.
Right. It causes a severe respiratory illness with a staggering fatality rate of up to 40%. And as of right now, we have 0 approved vaccines or cures for it.
It is a genuinely frightening pathogen.
Yeah.
But, this new research gives us the most detailed blueprint we've ever had of the virus's surface armor.
So the mission for our deep dive today is to understand exactly how scientists pulled off this structural mapping.
Right. We're going to explore how they bypassed extreme biosafety hazards using a really clever decoy system, how they used molecular supermicroscopes to map the virus down to the atomic level, and how they uncovered its secret mechanisms for infecting human cells.
And most importantly, we'll look at how we might finally use this blueprint to build a vaccine that stops it dead in its tracks. Okay, let's unpack this, starting with the sheer logistical nightmare of just trying to look at this thing.
Yeah, you can't just put this on a glass slide.
Exactly. Because Andes virus is so lethal, it requires biosafety level 3, or BSL-3, containment. For anyone who hasn't been in one of those labs, we are talking full hazmat suits, negative air pressure, heavily restricted access. It's incredibly slow, difficult work. You can't just casually toss a lethal virus under a microscope to study its native structure.
You really can't. So the researchers had to get creative. To study the viral armor, which is made up of 2 surface proteins called GAN and GC that form a protective lattice on the outside of the virus, they needed a safe workaround.
A decoy, essentially.
Right. They needed a model that could be handled in a standard BSL-2 lab. Initially, they tried using a surrogate virus, specifically a recombinant vesicular stomatitis virus, or RVSV.
Oh, I remember reading this part.
Think of this as taking a harmless livestock virus, hollowing out its dangerous parts, and essentially pasting the Andes virus armor onto its surface.
Mm-hmm.
But they ran into a fascinating physical problem.
Right, because the harmless RVSV virus is naturally shaped a bit like a bullet. Normally, if you put something like the SARS-CoV-2 spike protein on it, those spikes just coat the entire bullet evenly.
Yeah, they distribute everywhere.
But when they pasted the Andes virus proteins onto it, the proteins flat out refused to spread out. They specifically clustered only at the tightly curved tips of the bullet, leaving the flat sides completely bare. Why does it do that?
It's so interesting because it tells us something profound about how the virus builds itself. This polarization happens because the Andes virus proteins aren't just passive armor. They're active architects.
What do you mean by active architect?
Well, in a real Andes virus, there is no rigid internal skeleton giving the virus its shape. The surface proteins themselves physically pull the cellular membrane into a curve so the virus can bud out of the infected cell.
Oh, wow.
Yeah. On the RVSV bullet, the only places with enough natural curvature to satisfy these demanding proteins are the very tips.
That is an incredible insight into how the virus physically bends its environment. But practically, it meant the bullet virus didn't give them a dense enough coating of proteins to get a good structural map.
Right, it was too sparse. So they had to pivot to a different BSL-2 safe solution, virus-like particles, or VLPs.
So think of VLPs as hollow, empty virus shells. They have the authentic Andes armor on the outside, but absolutely no dangerous genetic instructions on the inside.
Exactly. They are totally safe to study. But historically, they are notoriously difficult to produce in large enough quantities for structural mapping.
Because the cells just don't pump out enough of them naturally.
Right. To fix this, the scientists bioengineered a solution. They added a specific molecular tag, an EVLP tag, to the viral proteins. This tag acts like a homing beacon that hijacks the host cell's own internal machinery.
The ESCR2 pathway, right. The endosomal sorting complex is required for transport.
Yes, the ESCRT pathway. It's essentially the cellular machinery responsible for pinching off membranes.
So by hijacking this natural pinching system with their engineered tag, the scientists tricked the host cells into mass-producing these hollow decoys.
Exactly. It boosted the yield of these safe particles by 30-fold.
A 30-fold increase. It is wild to think about the hurdles here. Because the real virus is basically a bioweapon, they had to hijack the cell's own pinching machinery just to print enough hollow decoy shells to look at it safely.
And that massive boost in yield really unlocked the whole study.
So just to do a quick recap here for you listening, we started with a massive BSL-3 danger. The scientists solved it using a VLP decoy system, hijacked the host cells to get a 30-fold yield increase, and discovered that these viral proteins demand extreme membrane curvature, which is why they cluster at the tips of other viruses.
A perfect summary of step 1.
It makes you wonder, if you were designing a vaccine, how might the clustering of these proteins at the extreme curves of a virus change how the immune system ultimately recognizes them?
That is a vital question.
Yeah.
And it's exactly what drives the need for high-resolution mapping.
So let's talk about that map. Thanks to the massive yield of those decoy particles, the team could finally use single-particle cryo-electron microscopy, or cryo-EM.
Which is an amazing technology. It involves flash freezing the samples in a miraculously thin layer of ice to stop all molecular movement, and then taking millions of snapshots.
Through this, they achieved something historic, a 2.35 angstrom resolution map of the Andes virus surface proteins directly in the membrane.
That resolution is just mind-blowing for a membrane protein.
Just to ground that measurement for a second at 2.35 angstroms, we aren't just looking at the overall shape, we are looking at the molecular scaffolding.
Right, it's a whole different level of clarity.
It's like upgrading from a blurry satellite photo of a city to seeing the exact layout of the individual bricks in a fortress. The researchers were able to see individual water molecules acting as structural support.
And specific sugar chains called glycans, specifically N138 and N350, that act exactly like physical clamps to hold the whole viral armor together.
The level of detail is staggering. And more importantly, it led to a massive course correction in the field of virology.
Yes, this is a huge turning point. You see, previous models of this viral armor were built by piecing together lower resolution images with crystal structures of isolated protein fragments.
But when you only look at isolated fragments outside of their native membrane, you make assumptions.
Exactly. This new ultra-precise map revealed that the previously accepted model of the virus's surface had a 30-degree rotation error.
30 degrees? That is a massive discrepancy at the molecular level. Imagine trying to design a key for a lock, but you don't realize the entire internal lock mechanism has been installed sideways.
It's a huge blind spot.
For years, scientists might have been trying to design drugs for a 30-degree phantom.
But that's a perfect analogy. Because of that 30-degree error, previous models completely mismatched how the top of the protein connected to its base.
They were basically plugging the wrong puzzle pieces together.
Right. The old model entirely missed two vital connection points between the structural proteins. With the new map, we can clearly see the three main pillars holding the viral fortress together.
Let's break those down.
You have a primary clamp, which is the GN1-GC1 interface, relying heavily on those sugar chains you mentioned. Then a secondary connection between neighboring protein heads, the GN1-GN2 interaction. And finally, a newly discovered third interface, the GN2-GC1 interface.
And that third interface features what the paper calls a reactive groove. I picture it like a deeply buried, highly sensitive pressure pad where a specific part of the protein rests.
That's a great way to visualize it. The precision of this new map finally shows us how tightly woven these proteins are and how they physically stabilize each other right up until the exact moment they need to deploy and infect a cell.
Okay, quick recap of Section 2. We got a 2.35 angstrom high-res map. That map corrected a massive 30-degree historical error in how we thought the virus was built. And it revealed three vital structural interfaces that hold the viral armor together.
Spot on. Which brings us to a really crucial question for you listening. Knowing that researchers had the fundamental architecture wrong by 30 degrees for years, how much does that level of atomic precision matter when we start building actual medical countermeasures?
It seems like it changes the entire game board. And understanding that exact corrected architecture is what lets us understand the virus's most devious weapon, its spring-loaded trap.
This is where the biology gets really cinematic.
It really does. Let's set the scene for how this infection actually happens. The Andes virus has just been swallowed by a human cell. It is now trapped inside a microscopic cellular garbage disposal called an endosome.
Right, the cell's defense mechanism.
The human cell thinks it has captured the invader and starts pumping acid into the endosome to digest it, but the virus is waiting for exactly this moment. As the environment becomes highly acidic, the virus has to execute a breakout.
What's fascinating here is the precise molecular chemistry the virus uses to escape. The researchers analyzed those tight structural interfaces we just talked about and found very specific amino acids called histidines hidden right at the critical connection points of the armor.
And they named a few specific ones, right? His 279, his 285, his 716.
Yes. And histidines are famous in structural biology because they act as perfect pH sensors. At a neutral pH, they don't have an electrical charge.
So they're just sitting there completely neutral.
Exactly. But when that acid floods the endosome, the environment fills with protons. The histidines grab those protons and suddenly gain a positive electrical charge.
Wait, so the virus intentionally places these histidines right next to other amino acids that already have a positive charge, like lysines?
Yes, exactly.
If you suddenly have all these new positive charges crammed into one tiny space when the acid hits, what happens? Does it just act like the two positive ends of a magnet repelling each other?
That is exactly the mechanism. It creates a massive, violent electrostatic repulsion. When the acid hits, the positive magnets in the viral armor repel each other so forcefully that the entire protein fortress blows apart.
So the armor literally shatters.
It shatters, and the underlying fusion protein GC springs forward like a biological harpoon. It plunges into the membrane of the endosome, pulling the virus and the cell together until they fuse.
The trap is sprung, the genetic material enters the cell, and the infection begins. That is brilliant.
It really is.
So the cell's own defense mechanism, the acid, is the exact trigger the virus needs to deploy its harpoon. It's a Trojan horse strategy. You're watching the virus use the host's own biology against it. Let's do our quick recap here. The virus enters the endosome. The cell pumps in acid. Those specific histidine proteins sense the acid, gain a proton, and act like repelling magnets. This blows the armor apart and fires the fusion harpoon into the cell.
That covers the whole mechanism.
So here is a question for you to ponder. Knowing that the virus relies on this highly specific, delicately balanced acid trigger to survive, how could we intentionally use that against it?
That is the ultimate goal for therapeutics. And to design those therapeutic specifically engineered antibodies, we have to look at how the entire viral shell behaves.
Right, zooming out from the individual proteins.
Exactly. The researchers zoomed out to look at how these individual protein fortresses link together to form the whole virus. What they found is that these viruses are not perfect rigid spheres. They are pleomorphic.
Meaning they have irregular curving shapes?
Yes. And to accommodate that curving shape, the armor has to bend. The researchers captured the armor naturally flexing at angles of 22, 25, and even 27 degrees.
So neighboring tetramers are connecting via GC-GC interactions, wrapping around the viral membrane like flexible hinges.
Right. And here's where it gets really interesting, because that flexibility introduces a huge mechanical problem when we try to attack the virus with antibodies.
Okay, break that down for us.
The researchers studied a very promising pan-hantavirus antibody called ADI-65534. Because they had their safe high-yield decoy system, they could map exactly how this antibody binds to the whole viral lattice at a stunning 3.3 angstrom resolution.
And they saw exactly how it locks on. The paper mentioned a specific mutation, G100AV, that perfectly locks on to the virus's GLN761 residue.
Yes, it binds beautifully to that specific target. But there is a catch. Standard human antibodies, like the IgG1 type, look like the letter Y.
Right, a classic Y shape.
The two top arms of the Y are what grab onto the virus. If an antibody can grab two different protein blocks on the virus at the same time, essentially holding hands across the gap, it locks the virus's armor in place so it physically cannot spring that acid trap.
But there is a literal distance problem here. The researchers measured the gap between two of these protein blocks, and it is 30 angstroms wide.
Which is a massive gap at this scale.
And the standard hinge of an IgG1 antibody can only stretch 24 angstroms. It literally cannot reach across the gap. It's too short to hold hands.
Precisely. Unless the antibody bends its arms backwards to an extreme 187-degree angle, which is biologically highly unlikely, it just can't reach.
So it can only grab one side, which isn't enough to lock the armor down.
Right. But because the researchers now understood this exact physical limitation thanks to the high-resolution map, they engineered a brilliant fix. They took the grabbing arms of the antibody and swapped the standard backbone for an IgG3 backbone.
And the reason you swap to an IgG3 backbone is because it naturally has a much longer, more flexible hinge region, right?
Exactly. By giving the antibody a longer structural hinge, it could comfortably bridge that 30 angstrom gap, securely locking two protein blocks together simultaneously.
And what was the result?
When they tested this engineered longer hinge antibody. It resulted in a threefold increase in its power to neutralize the virus, just by solving a mechanical distance problem.
That is incredible. Okay, time for a quick summary. We learned the viral lattice flexes to form its shape. We mapped the ADI 65534 antibody binding, discovered the 30 angstrom gap was too wide for standard antibodies, and fixed it by engineering a longer IgG3 hinge, boosting neutralization by 3 times.
Perfectly summarized.
It really makes you realize that biology at this microscopic level is just incredibly complex mechanical engineering. We're talking about measuring gaps and extending hinges to build better biological clamps.
It completely reframes how we think about drug design.
So if we can engineer the hinges of antibodies to fit the exact geometry of a specific virus, what other structural limitations in modern medicine might just be engineering problems waiting to be solved?
It's a huge frontier, and the final phase of this paper took all of this incredible structural knowledge and actually tested it in the real world inside animal models to see if it could translate into a functional vaccine.
Let's talk about those trials. They moved into mouse models using a vaccine platform called lion-rep RNA.
Right, lipid inorganic nanoparticle delivery of replicating RNA.
For those familiar with the recent mRNA vaccines, this rep-RNA is a self-amplifying version. Once it gets delivered into the cell, it actually makes copies of itself, theoretically giving you a much louder alarm bell for the immune system to react to.
Exactly, a stronger signal.
They tested a few variations, comparing a vaccine based on the natural wild-type Andes virus against a vaccine using the highly expressing decoy version we talked about earlier, the one with double mutations DM that produced 30 times more protein.
Right.
You would naturally assume that blasting the immune system with way more protein equals a much better immune response, wouldn't you?
You would certainly assume that, but the data showed a fascinating twist. When they analyzed the blood of the vaccinated mice over a nearly 2-month period, the high-yield DMEVLP decoy vaccine did indeed create significantly more overall antibodies that could bind to the virus.
But this is a massive, but it did not create more neutralizing antibodies than the standard wild-type version.
Right, the neutralizing levels remained completely flat.
Let's clarify that difference for a second because it's vital. A binding antibody just sticks to the virus. I like to think of it as a sticky note. It flags the virus for the immune system, but it doesn't necessarily stop the virus's machinery from working.
It's just a marker.
Right. A neutralizing antibody is entirely different. It acts like a piece of metal jammed directly into a lock. It binds to exactly the right spot to physically prevent the virus from springing its harpoon trap and infecting the cell.
That is a great distinction, and this connects to a huge, broader concept in modern vaccine design. Sheer quantity does not automatically equal quality.
It's all about where it binds.
Exactly. Just because the immune system is exposed to a massive amount of the viral decoy doesn't mean it learns to make the exact right type of weapons. In this specific self-amplifying RNA format, flooding the system might expose parts of the protein that actually distract the immune system away from the critical neutralizing targets.
So what does this all mean?
It means we have to be smarter, not just louder.
Let's do our final section recap. In the vaccine trials, they compared a wild-type rip-RNA vaccine against the high-yield DMEVLP decoy. The decoy created way more overall binding antibodies, but the actual neutralizing virus-stopping power was the same. Quantity didn't beat quality.
Right. It proves that just throwing more protein at the wall to see what sticks isn't the silver bullet.
If sheer volume isn't the answer, what exact part of the virus should the next generation of vaccines force our immune system to focus on?
The answer definitely lies in those tight interfaces and that spring-loaded mechanism we mapped out today. We need to stabilize those specific vulnerable regions so the immune system has absolutely no choice but to target them.
Make the lock the only thing the immune system can see.
Exactly. But let's push beyond the scope of this paper for a second and look at a really provocative, forward-looking concept.
I love these. Lay it on us.
We now know, with atomic certainty, that the Andes virus relies entirely on a perfectly timed spring-loaded trap triggered by those specific histidines sensing an acid drop in the endosome.
Right, the Trojan horse acid trigger.
What if we didn't even wait for the body to produce antibodies? What if we could design a harmless, inhaled molecule like a simple nasal spray that artificially drops the pH locally right at the surface of the virus while it is still sitting freely in the fluid of your lungs long before it ever gets swallowed by a cell?
Oh, wow. So you just spring the trap early?
Yes. Could we trick the virus into springing its trap prematurely? If it fires its harpoon into empty space, its complex armor shatters. It would be permanently disassembled and completely disarmed before it ever touches a single human cell.
We could essentially turn its own brilliant, evolutionarily perfected escape mechanism into its Achilles heel. That is a phenomenal thought to leave on, literally disarming the bomb before it's even brought inside the building.
It's the ultimate preemptive strike.
Thank you for joining us on this deep dive into the microscopic, mechanical world of the Andes virus. Keep questioning the hidden architecture of the world around you, because as we've seen today, sometimes the difference between life and death comes down to a few hidden atoms, a corrected 30-degree angle, and the engineered length of a microscopic hinge. We will catch you on the next deep dive.